All right, thanks everyone. This is the last you'll hear about Coral Ariza, hopefully for the rest of this conference, so I promise it'll be over quickly. I'm gonna talk about some recent work we've been doing on integrative species delimitation in North American mycoheterotrophic orchids. Okay, so orchids are a challenge taxonomically. Most of you know this in this room, but leafless parasites are also a huge challenge. These include holoparasites, so plants that are parasitic directly on other plants, and as John would call them, holomycotrophs or full mycoheterotrophs or whatever you want to call them. These are leafless obligate parasites of mycorrhizal fungi. They're difficult because they lack most of their vegetative tissues, so they lack leaf laminae. Some species actually lack roots, and so they only have an underground rhizome. And this lack of variation, or excuse me, this lack of features extends to the genome. And so Sean talked earlier about plastome reduction. Well, we had a lot of talks about that today. But you know, there's a relationship between, of course, the number of genes and the length of the plastome. If we're talking about plastid genomes, the amount of information that's available for reconstructing phylogenetic relationships, for taxonomic placement, and of course for species delimitation. And of course this extends to other genomes as well, like the nuclear genome. There are losses of certain gene families, mostly photosynthesis associated, but this is a real issue that continues to kind of plague us for those who study leafless parasites. Okay, so integrative species delimitation. Basically, this describes the process of analyzing multiple data streams in species delimitation. So this could include molecules, so DNA sequences, morphology, behavior, ecology, phenology, et cetera. So integrating multiple data sources. And we can make a further distinction between what's called iterative species delimitation and what I would call truly integrative species delimitation. And so if you do a Google search for integrative taxonomy or integrative species delimitation, you come up with thousands of hits. So it's very popular. So there's something to this. People are coming around to the idea that having multiple sources of data is generally a good thing in taxonomy. And so to make this first distinction, we can look at what I would call iterative species delimitation. So analyzing different data streams separately. So say we build a phylogenetic tree with DNA and then we layer morphology, behavior, ecology on top of that. You can take kind of a strict or a more relaxed approach to how you interpret those data. So you can require that all those data sets are congruent with one another, or you can relax that assumption and say, you know, so two of them have to be congruent or something, right? But that's what I would call iterative species delimitation, as opposed to what I would consider truly integrative species delimitation when we're analyzing all of the data in a single framework. For example, a really popular one is called IBPP. This is an extension of a multi-species coalescent method that's based on molecular data. So it includes multiple sequence alignments from multiple loci, but it also incorporates phenotypic or even ecological data under a Brownian motion model to try to improve estimates of species delimitation. And it has actually been shown to do that in both simulations and with some real data sets. So this would be an example of what I would consider truly integrative species delimitation. Okay, so in order to delimit species, you need to have a species concept. And when it comes to integrative species delimitation, the one most commonly relied on would be the general lineage or the unified species concept, which was published as a series of papers by De Caro's over the last quarter century. And the main idea here is that De Caro's equates species to separately evolving metapopulation lineages, right? So the word lineage is gonna come up a lot in this talk. Okay, but there's a problem. More recent studies have suggested that specifically the use of multi-species coalescent models, while they're great and everything, delimit population structure and not necessarily species. And so really this could potentially lead to oversplitting and really, like, is each one of these lineages a true species, or how do we define it as a species? That's the real question here. And so are lineages necessarily species? is the question we kind of want to address. I think the title of this paper sums it up, or at least how some of us feel. Lineages are not enough. And so the idea here is that, a lineage, if you really think about it, is a historical construct, and it's also kind of a snapshot in time, right? So if we're studying extant species right now, we're getting a glimpse of what they look like at this point in time, and we can go back in history, but we can't really go forward to say, like, if two lineages are separated for 10,000 years, what's to say in another 5,000 they can't come back together and fuse and form a single lineage? Okay, so the idea here is that this goes back to Simpson's evolutionary species concept and Van Valen's ecological species concept from the 1970s. The idea is that Simpson had a meaningful way of chopping up lineages built into his evolutionary species concept, and that was a change in role. So we're going to kind of focus on that word here. What is role? Well, really, role is referring to the ecological niche of an organism. So there's going to be a requirement of species having a somewhat distinct ecological niche or role, right? So a species is a lineage or a group of connected lineages with a distinct role. And the idea here is that, there's a growing group of taxonomists that want to, or think species should be something more than a historical construct or a historical pattern, and that there should be some kind of functional or ecological definition to species. Because if we think about biodiversity, if we think about functional ecology, those are going to be useful metrics, whereas otherwise we would just kind of be recognizing genomic lineages. Right? And so the problem is it's kind of hard to define ecological niche, i.e. role. And so we have to have kind of an objective way of doing this, or at least a way of leading us to hypotheses of role shift. And so the requirement here is that any fixed change in expressed organismal properties provides evidence for a hypothesis of role shift. And then if you've identified that, you could take a closer look and see whether, in fact, that this does, this, say, phenotypic change or physiological change that's fixed in two different populations really does have ecological meaning. Okay, so talking about North American mycoheterotrophic orchids, and specifically I'm referring to fully mycoheterotrophic or holomycotrophic orchids. They belong to 3 genera that are found in North America. The first used to be called Hexalectris, but a few years ago it was lumped into the genus Bledia based on a recent phylogenetic study. Hexalectris used to refer to the leafless species, of which there are about 9 in the bigger genus Bledia. Another genus that we've heard probably too much about today is Corallarhiza. You've seen plenty of pictures. I'm going to focus on again, Corallarhiza striata, the complex in the middle here. So you'll hear a little bit more about that, and then we'll be done with it. And then there's one species of Cephalanthera, a largely old world genus. This species, Cephalanthera austinii, is endemic to the Pacific Northwest and California, Oregon, Washington, gets just up into British Columbia and Canada. It's called the phantom orchid for obvious reasons. Really cool. Okay, so 3 genera. We're going to focus on the Corallarhiza striata species complex. Okay, so here is a general distribution of the complex from southern Mexico up to western Canada, across the Rocky Mountains, Great Lakes, and into the Canadian Maritimes. John just talked about the Corallarhiza striata complex. You have Corallarhiza bentley here, known from about 12 populations in eastern North America, so very rare, I would argue endangered. And then Corallarhiza involuta, about which we know very little in southern Mexico. And then we have this kind of widespread variable thing that I'm going to call Corallarhiza striata sensu stricto. That's all the way from Mexico up into Canada, and it's quite morphologically variable. Okay, so earlier work using integrative species delimitation from over 10 years ago basically allowed us to identify 3 species with pretty high confidence within the complex: Corallarhiza bentley, Corallarhiza involuta. Back to your question, Sean. Yes, these do have a very selfing kind of morphology, so reduced or even cleistogamous flowers, and even before the flowers open, sometimes the ovaries are fully developed and they're producing seeds. But then we have this Corallarhiza striata sensu stricto thing that we're going to focus on for the rest of this talk. And there are currently 2 described varieties, variety Verlandii in the southwestern US and Mexico, variety striata in the northern US and into Canada. And John was really talking about the variety on the right with the large showy flowers. Okay, so earlier work based on plastid genome sequencing. So let's see, these are whole plastid genomes. Basically identified 3 plastid lineages, right? So one corresponds to variety striata, another to, this is the large flowered, probably outcrossing species. This is variety Verlandii, another lineage to this, in the southwestern USA and Mexico. Probably more prone to selfing, as I'll show you here in a minute. And then this other lineage endemic to the Sierra Nevada in California. So this was kind of a surprise to us because these flowers, as I'm going to show you, kind of have an intermediate morphology between striata and Verlandii. Oh, and we also, collected populations in the Californian coast ranges and the Cascades of Oregon, all the way up to the Washington border. These kind of almost fall out at the base of variety striata, but we want to look more closely at those populations because they kind of resemble the other Californian populations morphologically. So in order to, well, the question, let me backtrack for a second. The question is, do these lineages actually represent separate species, or are we just looking at population structure here? And to help us address this question, we came up with ISSR-seq. This is a pretty straightforward method based on PCR amplification using microsatellite motifs as primers. And so you'd amplify several ISSR motifs across the genome. That stands for intersimple sequence repeat loci. So you're amplifying this higher complexity area between microsatellite repeats. And instead of scoring the bands, running them out on a gel and scoring the bands like people used to do back in the 1990s, this time we're sequencing them. So we're pooling them together and sequencing them. So this is a reduced representation method. And we're getting levels of variation that's quite similar to things like RAD-seq and genotyping by sequencing. And it's really easy. Undergraduates can do it in the lab. It's just PCR based. It's a lot of fun. Okay, so we're generating single nucleotide polymorphisms across the complex. Okay, so here's some results. Phylogenetic trees, a structure-like analysis, PCA. All of them really show a strong pattern of basically 4 lineages, right? So we're looking at something like 6,500 SNPs or single nucleotide polymorphisms. And we further did a Bayesian multi-species coalescent method called SNAP. And this allows you to test different number of species or different configurations of species. And the strongest evidence we received was for 4 separate species corresponding to each one of these lineages. So we have 4 separate species. No, just kidding. We have 4 lineages. So I'm going to talk about the integrative part after this, right? So we've identified 4 lineages. All right, so the first layer of data we'll talk about is morphology. Unfortunately, there are no nice discrete characters in this complex. Everything's continuous, so there's a lot of measurements and kind of looking at the patterns of morphometric variation in the flowers. Again, there aren't that many other characters as on these plants as a whole because they lack leaf laminae and roots. So you have to kind of rely on floral measurements. We did a principal component analysis of 14 morphological characters. And the first thing we see is a pretty clear separation of variety Verlandii in green, variety striata in red. But if we ask the question, is there evidence of fixed differences as required by our species concept? Yes, in some cases, if we look at individual characters like the labellum length, the length of the lip, Verlandii and striata have a pretty clear separation. If we're using like non-overlapping distributions of characters or character states or characters as a proxy of fixed differences. But then you have the individuals from the Sierra Nevada and the Coast and Cascade ranges. I should mention, because I failed to earlier, we did find a unique lineage in the coast ranges and Cascades, which is also a surprise to us. But they kind of occupy this intermediate position, so it kind of calls into question what this really means. And if you look at other characters, there's a lot of overlap. So generally speaking, we didn't really find evidence of fixed differences, at least in terms of floral morphology. So no evidence of fixed differences, i.e. hypotheses of role shifts here. Okay, so we also looked at phenology using flowering time of digitized herbarium records as a proxy for this. And we found a lot of overlap. We found some significant differences, but a lot of overlap. So probably no evidence of fixed differences there. We also looked at a proxy of reproductive biology by looking at the number of swollen ovaries on herbarium sheets and found that there was a significant difference between variety Verlandii and the other lineages. But again, there was a bunch of overlap here. Well, this means that largely Verlandii is probably selfing to a large degree, and then these others are probably outcrossing. We also did some ecological niche modeling based on abiotic data from the bioclim variables. And basically we found inconclusive evidence for differences among the 4 lineages. Essentially, these plants are finding similar habitats in different parts of North America and living there. So we didn't really find strong differences between abiotic variables. I think this is the 3rd time we've seen this slide today, but I'll just kind of walk you through it again. So we also looked at fungal hosts. And the first thing you can notice here is that there's definitely some clustering. So this is a fungal ITS tree, the striated complex. So the Sierra Nevadan individuals and the Coast Cascade individuals seem to be kind of using different groups of fungi. But again, there are some exceptions, and there's a lot of overlap between them. So again, no evidence for fixed character state differences, even though we found a statistically significant difference among each of the 4 lineages in terms of fungal host associations. Okay, and so the kind of pulling it all together, here we actually did some what I would consider actual integrative analyses of variation, looking at genomic lineages and what I'm calling the extended data, so morphology, phenology, fungal hosts, et cetera. And what we found was very strong evidence in all cases. This is a program called AssignPop, which I really like. The output is kind of like structure, but it's using machine learning to try to test different speciation scenarios or species delimitation scenarios. And we also conducted an analysis using redundancy analysis with variance partitioning, which allows you to test different numbers of species in your group based on the total data set combined and on each one individually. And in each case, we found evidence of 4 distinct species. But there's a problem. Based on our species concept, we haven't identified any fixed differences in any of the sort of extended data besides genomic data. And so to summarize, we found evidence for 4 genomic lineages. I'm stopping short of calling them species. But in terms of morphology, phenology, reproduction, abiotic niche data, we've basically failed to find truly fixed differences for any of these 4 lineages because we see overlap in pretty much all of them. And so we're going to kind of stop short and not call these separate species. I personally think that would be oversplitting. And so I think they're best probably recognized at the level of variety. Some may criticize us for being overly conservative here because there are conservation implications, et cetera. But at the end of the day, someone or something has to make a decision, whether that's a human or a robot. And I'm trying to be the human here and making a conservative decision as to avoid oversplitting. Okay, so more recent research is focused on sampling more intensively in contact zones. There are a couple of good papers in the last 2 years on this, so check those out. We're trying to do this right now, but it's really challenging for taxa with highly clustered distributions. These often have montane distributions and tend to be very, very clustered. We don't know why exactly. It probably has something to do with the fungal hosts, but we're trying to do this. We spent the last 2 years in the Pacific Northwest trying to collect as many populations as we could. We have some more field work planned for the Northern and Central Rockies this summer. And so hopefully we'll be able to do some more fine scale analyses where multiple lineages actually come together. So here in the Pacific Northwest and the Central or Northern Rockies. So to conclude everything, sampling in contact zones, we want to test the hypothesis that fungal host specificity is genetically determined in multiple species complex, including Corallarhiza. We're also developing a long-read bar-coded ISSR-seq protocol using PacBio sequencing, which would mean no more Illumina library preps, which would make the method even easier and cheaper. So more on that later. We're also doing this work in other complexes. Hana Thixton-Nolan, PhD student, is doing work in Maculata complex, and then we had a student working on Corallariza wistriana.
Excuse me, Greg.
And I'm done, thank you. Good timing.
Thank you so much. Time for questions.
Thanks, Greg. With this integrative view, I was surprised with everything you ran through that you didn't talk about geographic distribution as a difference in role. Number one, so why? And then secondly, I mean, traditional view is for species to coexist and therefore really to be species, they had to have ecological isolation, they had to do somewhat different things, and they had to have reproductive isolation. So, and then the final point is, and I say this with caution because I study adaptive radiations where this could be a problem too. Roles, say differences in ecological distributions, can be as much a product of the other species in a community as the individual species. So I wonder if you could respond to those.
Sure. First to the geographic bit. You know, we talk about sampling in contact zones. Part of the problem is we don't really know what's what yet. So the question is, are we going to see these sort of intermediate forms in these zones? Well, you know, genetically, morphologically, ecologically in terms of fungal hosts, or do they maintain distinctness in different populations or even within the same population? They're just hard to find. And you know, they're not ubiquitous, they're not randomly or uniformly distributed. So we're trying to focus on areas where what looks like this and what looks like this might be found in proximity to one another, which I think would go a long way in terms of determining whether they maintain distinctness in geographic proximity. And so, you know, looking at genomic data, looking at fungal host data on the ecological side, and looking at morphological variation.
Any other question? Really? Sure.
Who wants to ask a question?
Okay. Excuse me. Okay. Thank you so much, Greg. So.